during prophase 1, we see some similarities with mitosis. First of all, we're going to go from spaghetti form or chromatin to our packed up chromosome form. We are going to get rid of the nuclear envelope. Centrosomes in animals will appear, and then whatever kind of critter you are, you're going to have your spindle fibers are going to show up. If we stop this now, you wouldn't be able to tell if this cell was beginning the process of mitosis or meiosis. However, the next events are going to tell us if we're doing mitosis or meiosis. So the things that happen in prophase 1 that do not happen in mitosis are synapses. And in synapses, we are going to have homologous chromosomes come and associate with each other. So the chromosome 1 that you get from your dad is going to find the chromosome 1 that you get from your mom. Chromosome 2 that you get from your dad is going to come line up next to chromosome 1 that you get from your mom. Okay, that's synapsis. So the way that I remember it is that homochromos synapse together. Okay, so they're going to come and find each other. And when those homologous chromosomes are associated with each other, the next thing that they're going to do is they're actually going to remove segments of their DNA sequences and swap them with each other. So the resulting chromosome has a unique combination of genes that didn't exist on the same chromosome before. Okay, so crossing over, we're going to exchange genetic material of non-sister chromatids to create that novel genetic combination. Okay, so if this blue one is from the paternal line and the red one is from the maternal line, we're going to see dad's chromosomes swapping some genetic material with mom's chromosomes like we see here. Okay. What we do not see are sister chromatids swapping. If we had sister chromatids swapping, there would be no new genetic material because these are exact replicates of each other. These are just replicated DNA that are still stuck together. We're going to do it again, just bigger. And I will have this link to this video will also be in your Canvas shell. Okay. So here we've got our homologous chromosomes. These are sister chromatids. And these are non-sister chromatids. Okay. In crossing over, we have segments of non-sister chromatids swapping segments to create a novel combination of genes. Now, every chromosome can have a different pattern of crossing over. We might have multiple segments. So let me draw this up here real quick. So we could have multiple segments crossover. So we could have crossing over event here and here. We could have potentially a crossing over event like this. We might not, the homologous chromosomes might not do crossing over at all. So every pair of homologous chromosomes, every time that meiosis happens, we're going to have differences in how crossing over occurs. And this is then where our source of genetic variability comes from. Our first source was fertilization. Our second source of genetic variability comes from this crossing over. Okay, and crossing over occurs in prophase 1. So make sure that you write that down really big because that's an important concept for this. In metaphase 1, we are going to line up homologous chromosomes along the equatorial plane. So in mitosis, what we saw was our chromosomes lining up thusly. But in metaphase 1, we're going to see our pairs of homologous chromosomes lining up along the equatorial plane. Our third source of genetic variability in sexual reproduction comes from this independent assortment, because we might have our chromosomes line up in any number of ways. Okay. Each set of homologous chromosomes is going to be arranged independently of each other. So what we're seeing is that the chromosome set number 1 is not, the way that it lines up does not affect how chromosome set number 2 lines up. And that's true for each pair of homologous chromosomes. Okay. And we can actually calculate how many possible combinations of homologous pair combos there are. So in a diploid organism, we can take those 2 homologous pairs and raise that to the power of the number of chromosomes that we have. So in humans, we have our diploid organism, and we have 23 sets of homologous chromosomes. So when we put that into our math machine, that just from independent assortment alone, not considering crossing over, not considering fertilization, just crossing over alone, there's over 8 million combinations of possible ways that these chromosomes could line up along the equatorial plane during metaphase 1. Huge amount of genetic diversity coming from meiosis. All right, so for our anaphase, we are going to separate things. That's what anaphase do. But we're going to see some differences that we compare to mitosis. So again, in anaphase of mitosis, we saw that sister chromatids separated, something like that. Okay, but because back in metaphase, just one slide ago, we didn't line up our chromosomes. We lined it up homologous pairs. So now, during anaphase, we are separating homologous chromosomes. Those homologous chromosomes are being pulled to the opposite sides of the poles, and now these homologous chromosomes are no longer associated with each other, and therefore we have had our reduction event, and now we have gone from being diploid now we have reduced that number to be haploid. Okay. So the phase in which cells become haploid is anaphase 1. Anaphase 1 separates homologous chromosomes. Then we have telophase 1, and this looks very similar to as it did in mitosis, right? So we are going to have our chromosomes relax back into chromatid. So we're going from Cheeto form to chromatid form, which is, we've called that spaghetti in the past. Okay, so we're relaxing that back down. Sometimes we'll have cells that will reform their nuclear envelope. Sometimes they don't. Cytokinesis is going to occur, so we're going to take that cell juice and split it into 2. So now the question is, for you guys, are those sister chromatids identical in those 2 resulting haploid cells? And the answer is not necessarily, because crossing over back in prophase 1 gave us a gene shuffle. Those non-sister chromatids were reshuffled. We have a unique combination of alleles on our sister chromatids. So what we see, if we look at just these two sister chromatids, they're no longer identical because of that crossing over event. So at the end of meiosis I, we have two haploid daughter cells. So these two haploid daughter cells may or may not go into a process that looks similar to interphase. So in a typical cell cycle, after our cells are done replicating, we would have them enter G phase, right? They'd grow, we'd synthesize DNA, we would have our second growth phase. But during this modified interphase between meiosis I and meiosis II, we do not synthesize DNA. So then the question that you guys need to be thinking about is, why would replicating DNA between meiosis I and meiosis II defeat one of the purposes of meiosis? And now we have to remember what the purposes of meiosis are. There's two of them. Okay. One of them is to create genetic variability, and we did that with crossing over independent assortment. The other purpose of meiosis is to reduce the amount of genetic material by half, to go from diploid to haploid. Doubling the amount of DNA now would undermine the purpose of reducing our genetic material.